Assalamualaikum warahmatullahi wabarakatuh and good morning to examiner. Today I will talk about my finding on endophytic fungi from grasses, Poaceae, and their pathogenic ability. For introduction, endophytic fungi is microfungi that internally infect plant tissue and live with the plants for at least a part of its life cycle without causing any symptom. Endophytic fungi is ubiquitous in plant, and mostly of its member is from Ascomycota, but some of its member are also from Basidiomycota, Zygomycota, and Oomycota. Endophytic fungi is able to turn to pathogen under certain conditions such as temperature and become a pathogenic to host and non-host. Poaceae or grass. Grass is nearly ubiquitous family of monocotyledonous angiosperm or flowering plants. It comprises of 11,500 to 12,000 species. Poaceae or grass is ecologically and economically important. Grass is ecologically important because it can become a shelter and also can prevent soil erosion. Grass can be economically important due to the fact that cereal crops such as wheat and rice is in this grass family. Lawn grass, or its scientific name Azonophus compressus. Lawn grass is a creeping perennial that forms dense mat. It inhabits wetlands or moist to dry woodlands, disturbed areas, lawns, and cultivated fields. Lawn grass have ecological and medicinal interests. The ecological interest of lawn grass that it can be planted along the river dam to prevent soil erosion, and the medicinal interest of lawn grass is that the extract from the leaf can become a suppressive agent of sugar in blood. The objective of this research is to isolate and identify endophytic fungi from lawn grass, Azonophus compressus, and also to determine the pathogenic ability of the endophytic fungi towards the host and non-host plant. For materials and methods, the first step is the sample collection. For sample collection, 4 locations has been chosen as a location of the sampling. For each location, 5 healthy leaves is collected. The, the sample is then bring back to the lab and undergo a surface sterilization. Surface sterilization is done by using 70% ethanol, 5% sodium hypochlorite, and distilled water with 1 minute each. Next, the sample that has been surface sterilized is undergo imprinted technique, where the sample will cut into 5 segments and then imprinted in one plate and then transferred into another plate for the growth. Is single spore isolation. The endophytic fungi that has been obtained from the imprinted technique will be undergo single spore isolation to obtain the pure culture of the endophytic fungi. For single spore isolation, the spore suspension is need to be prepared. To prepare spore suspension, the colony is scraped. The surface of the colony is scraped with inoculation loop and then put in the biju bottle. Once the spore suspension is prepared, one loopful of the spore suspension is taken by using an inoculation loop and strike on the PDA using zigzag formation. Next, once the pure culture of endophytic fungi is obtained, it will be preserved in stock using half-strength PDA. Next is slide preparation. For slide preparation, is using 2 techniques which are slide culture and also cellophane tape. For fungal identification, there are 2 features that is being observed for the fungal identification, which are microscopic and also microscopic features. For pathogenicity tests, from all of the 47 isolates that has been obtained, few representative isolate is used for pathogenicity test. The pathogenicity test is done by inoculate the representative isolate on the lawn grass leaf using wounded and unwounded treatment. For cross-pathogenicity test, the representative isolate is inoculated on chilies fruit using unwounded and wounded treatment. For result and discussion, the first representative isolate is identified as Colletotrichum gliosporioid species complex 1. This representative isolate has cottony blackish green with small granules over entire surface of colonies. The conidia was cylindrical with blunt end, and it comprised of one isolate. Next, this representative isolate is identified as Colletotrichum gliosporioid species complex 2. This species has a compact grayish colonies. The conidia of this species is cylindrical with blunt end, and it comprises of two isolates. Next, this representative isolate is identified as Colletotrichum gliosporioid species complex 3. This species has a cottony whitish colonies, with the conidia is cylindrical with blunt end, and it's comprised of four isolates. Next, this representative isolate is identified as Colletotrichum gliosporioid species complex 4. This species have compact grayish-green colonies, and its conidia are clavate with blunt end, and this species comprises of 8 isolates. Next, this species is identified as Colletotrichum gliosporioid species complex 5. This species have a compact grayish to white colonies. The conidia is cylindrical to clavate with blunt end, and it's comprised of 6 isolates in total. Next, this representative isolate is identified as Colletotrichum acutatum species complex. This species have cottony whitish colonies, and it also has a bright orange conidial mass around over the colonies. The conidia is cylindrical to clavate with acute end, and it comprises of 7 isolates. Next, this representative isolate is identified as Fusarium oxysporum. This species has floccus whitish with pale orange microconidia formed at the edge of the colonies. The macroconidia is straight to slightly curved with slight hook apical cells and foot-shaped basal cells. The microconidia is oval-shaped with non-septate, and it comprises 2 isolates. Next, this representative isolate is identified as Fusarium solani. This species has sparsely to cottony whitish colony. The macroconidia is slightly slender and straight, blunt and rounded apical cell, and poorly developed foot-shaped basal with 5 to 7 septate. The microconidia is oval with 1 septate or non-septate, and it comprises of 3 isolates. This representative isolate is unidentified until species label and referred as fungi 1. This species have a cottony whitish colonies. This representative isolate also cannot be identified until species label and referred as fungi 2. This species have cottony brown to yellowish colony. Lastly, this representative isolate is identified as Curvularia lunata. This species has compact blackish green colonies. The conidia is tuberculous, curved, ellipsoidal to obovoid, and basal cell is paler than the middle cells. The conidiophore is raised in a group of 2 straight and septate. This species is comprising 1 isolate. The Coleotrichum species cannot be identified until species level because the Coleotrichum species have same morphological characteristics. And also, the environmental condition can affect the formation of the microscopic and macroscopic features of Coleotrichum species. The fungi, the unidentified fungi also cannot be identified until species level because this species cannot produce the observable microscopic features that can be identified its until species level, and is known as mycelial sterile. Result of pathogenicity tests of endophytic fungi on leaf of non-grafts after 7 days of inoculation showed that all the representative isolate is non-pathogenic towards the leaf of non-grafts, where the disease severity scale show that wounded and unwounded treatment with 0 scale. Page 5.2 shows some of the leaf of the long grass that have been inoculated with some of the representative isolate, and it showed no symptoms or signs on the leaf. For cross-pathogenicity tests of endophytic fungi from leaves of lawn grass on chili fruits after 7 days of inoculation shows that 5 of the representative isolate is pathogenic toward the chili fruit. Coleotrichum glosporioides species complex 4, Coleotrichum glosporioides species complex 5, Fusarium oxysporum, Coleotrichum acutatum species complex, and Fusarium solani is pathogenic towards the chili fruit. The disease severity for the Coleotrichum glosporioides species complex 4 is 1, indicating a slightly severe infection. For Coleotrichum glosporioides species complex 5, it has a disease severity scale of 3, indicating a severe disease. For Fusarium oxysporum, the disease severity scale is 1, indicating a slightly severe disease infection. For Coleotrichum acutatum species complex, the disease severity scale is 3, indicating a severe infection disease. For Fusarium solani, the disease severity scale is 3, which means the disease is severe on the chili fruit. 3. It shows the chili fruit that inoculated with the representative isolate and observable symptom from the inoculation. For picture A, the Coleotrichum glosporioides species complex 4 form a brown and slightly sunken lesion. For picture B, Fusarium oxysporum produce brown lesion with distinct margin. For picture C, Coleotrichum glosporioides species complex 5 produce water socket and dark grey sunken lesion. For picture D, the Coleotrichum acutatum species complex produce pale brown with slightly sunken lesion. And for picture E, Fusarium solani produce colorless sunken lesion. 9 species of endophytic fungi were identified along with the 2 unidentified fungi. All representative isolates were non-pathogenic towards the leaf of lawn grass, but Coleotrichum glosporioides species complex 4, Coleotrichum glosporioides species complex 5, Coleotrichum acutatum species complex, Fusarium solani, and Fusarium oxysporum is pathogenic towards chili fruit. For acknowledgement, I would like to thank you to my late mates, my colleagues, my postgraduate students, and mostly to my research supervisor for being